This recording goes over the light reactions of photosynthesis. So I've drawn the chloroplast in the corner here, and in the photosynthesis pathway, the light reactions happen in these thylakoid membranes and use the thylakoid space. So that's the green stacks here is the grana, the thylakoids are the discs. So when I draw my membrane, I'm actually drawing the thylakoid membrane here. And within the membrane, I will draw some structures that we'll discuss shortly. It's a lipid bilayer made of phospholipids. Okay, and let's look at the real structure of this and then we'll come back. Okay, so what I've drawn here are these rectangles, and the rectangles are representing what we call photosystems. So I'll abbreviate that PS. This one's PS2, so let's remind ourselves it means photosystem. This is where the pigments are located. So I'll draw some pigments represented as dots. These are actually molecules. And so the green would be examples of chlorophyll pigments. And then we also have other pigments in the photosystems, less of them, but here would be some examples of maybe carotene. We would also have some yellows, so those would be xanthophylls. I've drawn two chlorophylls in the middle. These are going to be special chlorophylls. These have names. This one is called P680, and these two, this complex is called P700. And I should mention this is photosystem 2 and 1. It is the opposite, so this one was discovered first, so they called it photosystem 1. But it turns out there's a photosystem 2, and when we draw it, we follow the electrons from photosystem 2 over to photosystem 1. So there's our pigments, and if we look at pigments, you know, they are molecules that capture light. So let's look at what light energy is and how the pigments capture that. We said the part of the chloroplast that absorbs the energy is the photosystem. So these are representing chlorophyll molecules, or it could be carotenoids in there as well. And you can see the energy comes in the form of a photon, light energy, hits the chlorophyll, or the pigment, and the energy level bounces up. So the electrons are going to get energized. Then when they fall, they're going to transfer that energy to another pigment molecule. Those electrons energize, and then when they fall, they transfer it to another pigment, so it bounces around until it hits these two middle pigments here. They're special chlorophyll A's. That's the P680 or P700. When those electrons get bounced out of their orbitals and energized, they can actually get captured at a different part in the photosystem called the primary electron acceptor. And that primary electron acceptor captures those, and it's going to hold them there until it transfers the electrons to the next step. These are some examples of photosystems. They're real structures. This is the thylakoid membrane. And you can see there's lots of alpha helices in the membrane space. That's where the phospholipids are. Alpha helices tend to be where hydrophobic entities are, so that makes sense that they'd be embedded in the phospholipid bilayer. And then we can see there's parts that stick out, some beta-pleated sheets, beta barrels, and it's pretty complicated. So this is a monomer of photosystem 2 in this organism here. And then you put those together and you can actually, you'll look, it's actually a dimer, the real photosystem. So here there's one, and then there's the other, and both of them together make your photosystem. And then we also have in the violet, in this case, as well as here, those are all the pigment molecules. So you can see they're embedded in that membrane, and they're going to capture light. And this is photosystem 2 from a different organism. You can see it's also two monomers put together. And you can see, again, the chlorophyll all accumulated in the membrane, and the proteins as well. Light, or solar energy, behaves in two ways. One, it behaves as a wave. So there are different sized waves, or we call them different wavelengths. So some wavelengths are long, and some wavelengths are short. And the wavelength is really the distance between two crests or two troughs. So either one of those. So this is a long wavelength on the left here. This is a shorter wavelength. The longer the wavelength, the less energy the wavelength has. The shorter the wavelength, the more energy it has. Besides acting as a wave, light also acts as what we call photons, which are these discrete particles of energy. And so photons are what is transferring the energy into the photosystems in order to allow photosynthesis to work. The electromagnetic spectrum of different wavelengths runs from very high energy wavelengths over here with gamma rays. They're very, very small wavelengths. It means they're high energy all the way to radio waves, which are very long wavelengths, which means that they are low energy. In this electromagnetic spectrum, there's a part of it that's called visible light, and that's what we actually see as humans. Other organisms can pick up UV waves, sometimes infrared waves, but for us, we just see visible light. So visible light is, the Roy G. Biv idea. So the red is the longest wavelength, the lowest energy. Then we go orange, yellow, green, blue, and violet. And violet is the shortest wavelength, so that means it's the highest energy. So these wavelengths can be captured, in addition to infrared and UV, actually, by the pigments in chloroplasts. And the wavelength energy can be used in order to perform photosynthesis. Here's a picture of the chloroplast and the thylakoid membranes, and that's where the chlorophyll and carotenoids or other pigments are located. And when the light hits the chloroplast, there are three things that can happen. The light can be absorbed, the light can be transmitted, it goes straight through, or the light can be reflected. Any kind of transmitted light or reflected light, those are light wavelengths that we see with our eye. So we have our human eye over here, and that's what it sees. It sees the reflected, and it sees the transmitted light. Anything that's absorbed is not seen. And what that tells you is what wavelengths of visible light the pigments are able to capture. So in this case, if we see green, like the plant leaves are green because of the chlorophyll, the reason is because chlorophyll captures all the light except for the green wavelengths. Those go through or bounce off. So that's what we see. So if we had an object that looked blue, that would mean the wavelengths coming in, all of them would get absorbed except for the blue wavelength coming off. That would either go through, transmitted, or reflected. So the one that you see is the one that's not absorbed. Chlorophyll is the predominant pigment in chloroplasts, which is why the leaves look green, but there are also other pigments in there called carotenoids. They're important for capturing additional light energy for photosynthesis at different wavelengths than chlorophyll is able to, as well as capturing extra energy that's coming into the chloroplast that may damage the molecules there. So they do photoprotection to absorb the energy before it damages the chloroplast. This is an example of the different wavelengths of light versus the absorption of light by chloroplast pigments. And they've charted each pigment in turn. So chlorophyll A is one type of pigment molecule, and pigments are just molecules. It looks green-blue, and that's because it absorbs in the violet and indigo wavelengths, and then you can see it dips down, so it does not absorb in the blue or the green very well, and then it'll come back up in the orange and red, and so that means that it absorbs in the blue-violet as well as the red-orange. So that means it looks what it reflects, blue-green. Chlorophyll B is an accessory pigment, so it's a secondary chlorophyll. There's C and D as well. There's not as much of the B as the A. But it is also green, like all chlorophyll, but you can see it peaks in a different spot. It can still absorb in this range, but instead of reflecting blue, the chlorophyll B absorbs blue pretty well and really just reflects the green and yellow and then absorbs the orange. So chlorophyll B looks olive green. And then we get to carotenoids, which is a large category that includes the carotenes, it includes lutein, it includes the xanthophylls, the violaxanthins, so it's diverse. But overall, the carotenoids can capture wavelengths in this region, and they reflect all this. So they're going to be the ones that look yellow and orange. Carotenoids would appear yellow and orange because they capture the violet and blue wavelengths of light. Putting all these together, so different plants have different proportions of the pigment molecules, but if we put those together, we can get what we call an action spectrum. So this is an absorption spectrum. Here's our action spectrum, and you can see that all of these pigments absorbing in this region kind of get combined together to do photosynthesis. So this is a combination of all this absorption here. And then we see another peak down here, and that's because the absorption basically combines in order to be able to do a lot of photosynthesis. So plants are not going to be very good at doing photosynthesis in this green or yellow wavelengths of light. They are good at doing photosynthesis. They can capture that light in the blues and violets, as well as in the red, orange. So if you were to try to grow a plant in under one color light, you might pick violet light. That would work pretty well. Or you might pick red light, but you would not pick green light, because you can see the action spectrum is very low there, not a lot of photosynthesis, because the plant reflects the green. It does not capture it. This is actually an interesting experiment. Engelmann did this a long time ago. I wanted to see what wavelengths oxygen was produced at, which is a byproduct of photosynthesis. So what he did is he put this strip of algae, which is a photosynthetic protist, on a slide, and then he plated bacteria that need oxygen in order to divide and live. And so he started with all the bacteria just near the algae and waited. And he shined light of different wavelengths through a prism across the algae strip. So basically the idea is that whichever algae makes more oxygen, the bacteria should grow more, and you'd have more bacteria in that area. And that's what happens. This is how I found out that violet light and some blue is really good at producing oxygen, which means that photosynthesis is happening a lot because he has lots of bacteria. Green light, yellow light, not a lot of bacteria grew. Those must not be used for photosynthesis. And then again, we get to our oranges, and then particularly some red light. That again is good for photosynthesis. So you can actually see his bacteria growth curves look like an action spectrum when we can measure it with direct oxygen levels or other means. So it's a rather clever experiment without a lot of technology involved. Here's a picture of chlorophyll, and the things I want you to notice is the head is the part here that they've highlighted. The head is where the energy is captured. So the magnesium ion coordinating the nitrogens and carbons, this structure here is going to be good at capturing photon energy. So that's where the light is absorbed. So that's going to be where it absorbs the red and the violet and blue light very well. It's not going to absorb green. This kind of structure doesn't absorb green. So that's reflected. Chlorophyll A, the most common pigment, has a methyl group here, whereas chlorophyll B has an aldehyde group here. And so that makes them slightly different, which is enough to make them capture different wavelengths of light. Then you see this tail. So this is a hydrocarbon tail, carbons and hydrogens. And so that part makes sense because this pigment has to be put into the phospholipid bilayer. So the hydrophobic part interacts with the bilayer hydrophobic tails well, and so this can be embedded in the membrane through its hydrophobic tail and then have its little head stick out ready to capture light. When it captures light, the electrons in the different orbitals can get energized to the next orbital, and when that happens, they'll stay there for a short period of time, and then they'll fall back down to the more stable state. If there's nothing else around except for a single chlorophyll, that falling down will actually fluoresce. It produces a pink color under UV light. But if there are other pigments around, when it falls down, the other pigments can capture the energy and bounce their electrons up. Those fall down, and other pigments capture the energy and bounce their electrons up, and so forth. So that's how a chlorophyll molecule would capture the energy of photons and transfer it to the energy of the electrons, and then that can get transferred to heat and fluorescence or to another chlorophyll molecule. So back to our drawing. Alright, so we have light energy coming into the system. It's going to hit a chlorophyll molecule, and it's going to take that chlorophyll and bounce it out of its orbit and energize it, and then it's going to fall down and get captured by the next pigment molecule. Again, it gets bumped out of orbit and falls down over and over and over until it gets captured by these P680 chlorophyll molecules in the middle. So the chlorophyll molecules there are going to also energize, and the electrons are going to bump out of orbital, but this time there's actually an entity that can capture those electrons, and we'll trace 2 at a time, 2 electrons. And they are captured at the top here with this structure, which we'll call the primary electron acceptor. So it's the primary electron acceptor. So basically that's just part of the complex that can capture the electrons and hold them there for a little bit. The same thing is going on in our photosystem 1, second one in line. The energy of light is hitting, the photons are hitting the pigments. If they're the right wavelength, they get captured, and then they get bounced around until they get captured by these P700 chlorophyll A's. There's also a primary electron acceptor on this photosystem. It's going to capture the electrons at a high energy state as well. So where do the electrons go from there? Let's look at PS2. So the electrons are at a high energy state, and next to them is an electronegative complex. So this complex here, these 3, are part of a chain we call the electron transport chain, or ETC. So ETC equals the electron transport chain. And it's just what it sounds like. Electrons get transported from one of the complexes to the next. It's a combination of proteins and non-protein molecules. There are carriers in between that are embedded in the membrane as well, but for our purposes I kind of just like to draw it simply. So the electrons, because this complex is more electronegative than the primary electron acceptor, the electrons are going to fall down into this first entity of the electron transport chain, and they release a little energy. Then they're going to get pulled by the next member, so even more electronegative. So it's going to be able to pull those electrons towards it. The electrons lose a little more energy, and then they bounce to the last of the electron transport chain, and they've lost a lot of energy. So why do I mention that they're losing energy as they jump down, down, down the chain? That energy is harnessed in order to do work, and in this case it's a work of pumping hydrogen ions across the thylakoid membrane. So hydrogen ions are not going to go across themselves. They're charged. They can't cross a phospholipid bilayer, but with the help of the electron transport chain complexes, they are able to be pumped inside of the thylakoid, and they're going to really fill up that space. So you can see this going from low to high, which is active transport, and that transport comes from the electrons falling down the electron transport chain. So that's one important component. These hydrogen ions, this concentration gradient, is going to allow us to make ATP, which we'll look at in a moment. Back to the photosystems for now. So we got rid of the electrons from P680, an electron. So we actually are now with the P680 has lost an electron, so it's actually called P680 plus. That's its positive. Same with P700, P700 plus. And that's when it loses the electron. Now it needs to gain the electrons back in order to be refilled so that it can bounce the electrons out of the orbital again and keep this going. So where do we get the electrons to refill P680 plus? P680 plus is like a black hole for electrons. It's very, very electronegative, and so it can capture electrons from molecules very well. So in this case, remember our photosynthesis inputs and outputs. Here's our chloroplast. Remember, we have thylakoids. That's where this is occurring. And we have inputs and outputs. So in the light reactions, our input is light energy and water. And then our output from that light reactions is our oxygen. So water, H2O, carries electrons in the bonds. We're going to extract those electrons, and a way to do that is to break the bonds. So an enzyme is going to break the bonds between the oxygens and hydrogens. The electrons are released and captured by this P680 plus because it's very electronegative. So the electrons are going to get transferred, 2 electrons, since we're tracing 2 at a time, from the water into the P680 plus chlorophyll. What does that leave us with? That leaves us with some hydrogen ions, 2 of them per water. And it also leaves us with an oxygen atom. Now, an oxygen atom can't exist by itself in the system, but it can be held at the photosystem until another oxygen comes along. So what happens is you have a second water split. So you get 2 more hydrogen ions, and then you get another oxygen. Those 2 oxygens are held next to each other. An enzyme makes a bond between the 2, and now you have an O2 molecule. So that is your oxygen gas product. So all the oxygen that we breathe comes from plants or algae or photosynthetic bacteria. So the water is where we ultimately get our oxygen to breathe. This oxygen can go and be used in the cellular respiration within the plant or algae itself, or released to the atmosphere through those stomata pores on the bottom of the leaf, and those will go and be taken up by other organisms in order to do cellular respiration. So that's how we replenish PS2. What about PS1? Well, in this case, we have a nice little situation where our electron transport chain is sitting right next to our PS1, and it has electrons in it. And so what ends up happening actually is the electrons get transferred from the electron transport chain end into the P700 plus, and that's photosystem 1. And then you'll notice we needed to get rid of the electrons here in order to keep the chain going, because you always got to be shipping electrons out or else you back up the chain and it all stops. So in order to ship those electrons out, we're going to use an electron carrier molecule or an electron shuttle. So this is called NADP plus when it's empty, so I think of it as the cab. And then it picks up some electrons and it's going to go to being full. Pick up 2 at a time. There are little electrons, and now it has a different name, NADPH. So it picks up a hydrogen ion, but the most important thing is that it picks up 2 electrons. That's when the cab is now full. Then it's going to go deliver the electrons to another part of photosynthesis, and it's going to come back empty when it's lost its 2 electrons and its hydrogen back to NADP+. Continuous cycling, and being reused in the cell. So this is our electron shuttle, and note that the P, really it's nicotinamide adenine dinucleotide phosphate. You won't be required to write that on the exam, but you do need to remember that the phosphate one, the P, goes with photosynthesis. There's a similar molecule, NAD plus and NADH. You can see it's the same except for no phosphate. This is going to be for cellular respiration. So don't get confused. So the phosphate's important to remember. And really this cycle here is a redox cycle. Just like all these transfers of electrons, all the electron transfers are redox reactions. So gain of electrons, you're reduced, like Rig, reduction is gain. So you're reduced, and then you give up electrons, so now you're oxidized. And the next one's reduced. Then you give the electrons up and you're oxidized. Then the next one's reduced, and you can see that just keeps going. So if we look at NADP plus and NADPH, when we pick up the electrons, we gain electrons, that's Rig, G for gain, which means it's NADP plus is reduced. to NADPH. When the electrons go away, they're lost. Oil is oxidation, so NADPH is oxidized to NADP+. So let's look at our NADP+ in the context of our light reactions. So that's what's going to be, I'm going to put it here. This molecule is an enzyme, an electron transport chain combo. I'll just draw one of them, but there's a number of things here. It's called NADP+ reductase. ACE for enzyme, reductase because it reduces NADP+. And so then what we're going to do is drop the electrons into NADP+, so the electrons get transferred into NADP+. That is going to turn into NADPH. With those electrons in it, so now it's the full cab, so our 2 electrons, and those are going to be shipped, the molecule, the carrier will be shipped to the Calvin cycle. Then it's going to be embedded into the sugar that the Calvin cycle makes, or the carbon compound. So that's where the electrons come into play, and you can actually see that the electrons that will ultimately be embedded in the sugar originally come from water. Then we have one last part we need to look at. So I'll make this small. This is going to be why our hydrogen ion concentration gradient is important. To remind you, the point of the light reactions is to make molecules to run the Calvin cycle. So those two are the NADPH with the electrons, and we need ATP in order to be able to build our sugar. So this is where ATP comes into play. So I'm going to continue the membrane over here because it's easier to draw. And I'll draw a molecule in the membrane over here. Now all this stuff is repeated over and over and over in the thylakoid membrane. There's many photosystem, electron transport chain photosystem combos, and then there's many what we call ATP synthases, which are going to synthesize ATP and their enzymes. And those ATP synthases are spin around actually, and they're run by the hydrogen ions flowing through them. So there's, again, lots and lots of hydrogen ions being pumped into the thylakoid space. Very steep concentration gradient. The tendency of those ions is to spread back out. The only exit's through the ATP synthase. So the hydrogen ions are going to travel out the ATP synthase. The ATP synthase actually spins the bottom part here, and that spinning, they're not totally sure how it works, but what it ultimately does is takes ADP plus inorganic phosphate and puts it together to make ATP, the other essential product of the light reactions. So that product is also going to go to the Calvin cycle and then be used to make the sugar carbon compound at the end. So that is an overview of the light reactions. And I'll make them small. So what I think would be good is if you were to draw these processes out. I mean, it's useful to look at this, but the real benefit is to draw it out and see if you can trace and talk about all the processes that are happening. Just mark that there's an additional electron transport chain here that doesn't have any kind of pumping though. So that's the basic summary of your light reactions. This is a nice picture of the actual molecular structure of the photosystem 2, the electron transport chain here, photosystem 1, the little electron transport chain here, the reductase to make NADPH, and then here's your ATP synthase. You can see they have it spinning. This part's the part that spins, and that spinning is what takes the ADP and inorganic phosphate to ATP. The hydrogen ions, you see they're flowing through, and hydrogen ions are being pumped in this direction during the electrons falling down the chain. Note that the hydrogen ions are being transported into the lumen or the thylakoid space, and that's because they get concentrated there because it's small. So that's a general strategy. If you need a concentration gradient, pumping them into the small space is going to be most efficient to get them very concentrated. And so you'll notice that in photosynthesis and cellular respiration.